go.
In this video, I will be going through the wall structure of an alimentary canal organ and specifically giving you information on the histology of its 4 main layers. Now, the mindset that you need to have as you're thinking about this depiction is that this is essentially, like if you were using car buying terms, the base model. So if you went into the dealership and said, show me the base model for alimentary wall histology, it would be what I am going to put on the board here. As we go through the different alimentary canal organs, we'll talk about how each organ specializes one or more of these layers in order to make it do its job. So you can think of those as the kind of organ-specific upgrades that layer would have. Okay, so as far as our base model, what I have written up here for you right now is just the term lumen. So this is the location that the food material would be in, and it would also be where secretions would be added to that food material. Okay, so as I'm putting up this blue line here, and I'm going to make it kind of as simple structurally as possible. This layer that is next to the lumen would be the mucosa layer. Okay, and just a reminder that we have mucous membranes in the alimentary canal, so the entire length of the alimentary canal is going to be lined by mucous membranes, because mucous membranes are the type of epithelial membrane that we have in organs that are open to the outside. Okay, so this would also be the location where the epithelium would be located. So if I put this extra little layer here, this is where our epithelium would be located. Okay, and so remember, epithelium is a tissue that covers body surfaces and lines body cavities. Okay, so as we get to individual alimentary organs, we'll talk about what type of epithelium we have there and why that specific epithelium is there. Okay, our next layer, as we go past the mucosa, is the submucosa. Okay, and its name is telling you that it is deep to the mucosa, or further from the lumen. Okay, and this is going to be a loose connective tissue layer. It's going to have a lot of blood vessels, nerve plexuses, lymphatic vessels in it. There can be multicellular glands here, and there can also be collections of lymphoid tissue. The next layer is the muscularis externa. Sometimes it's just referred to as the muscularis layer. Okay, and this layer is composed of 2 sublayers of muscle. And remember, since this is a hollow visceral organ, the type of muscle that we're going to have here is smooth muscle. One of these layers is circularly arranged, so we can change the lumen size. The other is longitudinally arranged, so we can change the length of the organ. And by alternating the activity of these 2 layers, this is what allows us to have activities like peristalsis, one of our propulsive activities, and another activity called segmentation, which we'll see in reference in the small intestine as a way of mixing food material with secretions. Okay, our final layer is the serosa layer. Okay, so we have in many of our alimentary canal organs both a mucous membrane and a serous membrane, because serous membranes are found in closed body cavities, and many of our alimentary canal organs are found in the abdominal pelvic cavity. Okay, so any organ that is an alimentary organ that's in the abdominal pelvic cavity will have a serosa, and another name for this will be the visceral peritoneum. Okay, because the peritoneum is the specific serous membrane that we have in our abdominal pelvic cavity, and it's the visceral layer that is on the surface of the organ. Now, if you have an alimentary organ that is not in the abdominal pelvic cavity, like the esophagus, then you will not have a serosa, because you can only have a serous membrane where you have, or this particular serous membrane in the abdominal pelvic cavity, because that's the only place where it's found. Okay, so the esophagus, instead of a serosa, will have a substitution and a firmer, more dense connective tissue structured membrane, or layer, excuse me, which is referred to as the adventitia. Okay, so this is the base model for our histology of the alimentary wall.